cat sits on a wooden fence. The wind blows through its fur. The fur moves like waves. Some cats have short, flat fur. Other cats look like walking clouds. Their fur stands out in every direction. Why does this happen? The answer is not simple. Fluffy cats exist because of many reasons. Some reasons are easy to understand. Other reasons need years of study to explain. Scientists have spent decades watching cats. They study cat fur under microscopes. They map cat genes in laboratories. They track wild cats in mountains and forests. This is the story of fluffy cats. We will look at this topic from 7 different views. Each view gets deeper. Each view shows something new. A child sees fluffy fur one way. A scientist sees it another way. Both views are correct. Both views matter. The question stays the same at every level. What makes a cat fluffy? The answers will surprise you. Level 1. House cats. Some cats are fluffy. Some cats are not fluffy. When you touch a fluffy cat, your hand sinks into the fur. The fur feels soft. It feels thick. When you touch a cat with short fur, you feel the cat's body right away. A Persian cat is very fluffy. The fur on a Persian is long. It covers the whole body. The fur on the face is fluffy too. The tail looks like a big feather duster. When a Persian walks, the fur moves with each step. A Maine Coon is also fluffy. This cat is bigger than a Persian. The Maine Coon has long fur on its belly. The fur on its tail is very long. The ears have small bits of fur sticking out from the tips. Fluffy cats need help to stay clean. The long fur can get tangled. It can form knots. People use brushes on fluffy cats. The brush moves through the fur. It removes loose hair. It keeps the fur smooth. Fluffy cats come from cold places. In cold places, animals need warm coats. The thick fur keeps the cat warm. It works like a blanket. Air gets trapped in the fur. This trapped air keeps heat close to the cat's skin. Short-haired cats come from warm places. In hot weather, thick fur would make a cat too hot. Short fur lets heat leave the body. The cat stays cool. When you see a fluffy cat, you are seeing a cat built for cold weather. The parents of fluffy cats were also fluffy. Baby cats get their fur type from their parents. If both parents have long fur, the babies will probably have long fur too. Fluffy cats look different from other cats. But they are still cats. They play, they sleep, they hunt. The fluffy fur is just one part of what makes them special. Level 2. Outdoor cats. Cat fur has 2 main parts. The bottom layer is called the undercoat. These hairs are short and soft. They grow close to the skin. The top layer has guard hairs. These hairs are longer and thicker. They stick out from the body. Fluffy cats have both layers. The undercoat is very thick in fluffy cats. Persian cats have extremely dense undercoats. The guard hairs are long but fine. This combination creates a cloud-like appearance. Maine Coon cats have different fur structure. Their guard hairs are longer and stronger. The undercoat is thick but not as dense as a Persian. Ragdoll cats have soft guard hairs with medium undercoat density. Siberian cats developed in Russia. Russia has very cold winters. These cats have triple-layer coats. The undercoat is thick. The middle layer adds more insulation. The outer guard hairs repel water. Himalayan cats came from breeding Persians with Siamese cats. They kept the Persians' fluffy coat. Cat fur changes with seasons. In fall, cats grow more undercoat. This prepares them for winter. The extra fur provides more warmth. In spring, cats shed this extra undercoat. Large amounts of fur come loose. This process is called blowing the coat. Fluffy cats shed more fur than short-haired cats because they have more fur to lose. The fur helps control body temperature. In cold air, the cat fluffs up its fur. This creates air pockets. The air pockets trap heat. In warm weather, the fur lies flat. This lets heat escape from the skin. Fur type comes from genetics. Cats have genes that control fur length. When 2 fluffy cats mate, their kittens usually have fluffy fur. Sometimes a fluffy cat and a short-haired cat have kittens together. Some kittens may be fluffy, others may have short fur. The gene for long fur can hide in a short-haired cat. That cat can still pass long fur to its kittens. Different breeds show different amounts of fluffiness. Breeders choose cats with the fluffiest fur. They mate these cats together. Over time, the breed becomes fluffier. This is how humans have changed cat fur over many generations. Level 3. Working cats. The structure of cat coats divides into 2 categories. Single-coated cats have only guard hairs with minimal undercoat. Double-coated cats possess both substantial undercoat and guard hair layers. Most fluffy breeds are double-coated. The density and length of each layer determines the degree of fluffiness. Norwegian forest cats evolved in Scandinavia. The harsh climate selected for cats with water-resistant outer coats and insulating undercoats. The guard hairs contain more oil than other breeds. This oil sheds water and snow. Turkish Angora cats developed in the mountains of Turkey. Their coat is single-layered but very long. Without a dense undercoat, the Angora appears fluffy but feels lighter than double-coated breeds. Burman cats show moderate fluffiness. The coat is silky with less undercoat than Persians. British Longhair cats are the long-haired version of British Shorthairs. They have plush, dense coats with substantial undercoats. Somali cats are long-haired versions of Abyssinians. Their fur is finer and lies closer to the body. Geographic origin strongly correlates with coat type. Cats from northern regions developed thick coats for survival. Cats from Mediterranean and Middle Eastern regions developed longer but less dense coats. The temperature range in these areas is less extreme. The cats needed protection from sun and moderate cold but not severe winter conditions. Selective breeding has intensified natural coat characteristics. Early cat breeders noticed cats with unusually long fur. They bred these cats together. Each generation showed longer fur than the previous one. This process continued for decades. Modern Persian cats have much longer fur than the original Persian cats from the 1800s. Long-haired cats groom differently than short-haired cats. They spend more time licking their fur. The rough tongue removes loose hair and debris. However, the tongue cannot reach all areas effectively. Mats form when loose hair tangles with attached hair. These mats appear most often behind the ears, under the legs, and on the belly. Health issues accompany long coats. Cats swallow loose hair during grooming. This hair forms balls in the stomach. The cat must vomit these hairballs out. Fluffy cats produce more hairballs than short-haired cats. Skin conditions develop under matted fur. The mats pull on the skin. Air cannot reach the skin surface. Moisture gets trapped. This creates conditions for bacterial or fungal growth. Owners of fluffy cats must commit to regular grooming. Daily brushing prevents mat formation. It removes loose undercoat before the cat swallows it. This maintenance is necessary for the cat's health and comfort. Level 4. Breed standard cats. The genetic basis for long hair in cats centers on the fibroblast growth factor 5 gene. This gene regulates the hair growth cycle. A mutation in FGF5 disrupts the normal signal that stops hair growth. Hair follicles continue producing hair for an extended period. This results in longer individual hairs. The mutation is recessive. A cat must inherit the mutated gene from both parents to display long hair. Different mutations in FGF5 exist across breeds. Some mutations cause very long hair. Other mutations cause moderately long hair. Ragamuffin cats carry mutations that produce extremely long, dense coats. Balinese cats have a different mutation that creates long, silky hair without dense undercoat. The specific mutation affects both length and texture. Thermoregulation in long-haired cats involves complex mechanisms. The coat creates a boundary layer of still air next to the skin. This layer insulates the body. In cold conditions, the erector pili muscles contract. This makes the fur stand perpendicular to the skin. The coat thickness increases. More air becomes trapped. Insulation improves. In warm conditions, these muscles relax. The coat lies flatter. Heat dissipates more easily. Turkish Van cats developed near Lake Van in Turkey. The region experiences hot summers and cold winters. These cats have a unique coat adaptation. The undercoat is minimal in summer. It grows thick in winter. This seasonal variation is more extreme than in other breeds. Nebelung cats show consistent coat density year-round. They originated in cooler climates with less seasonal variation. Evolutionary trade-offs exist with long coats. Long fur provides superior insulation. However, it increases body weight. The cat must carry this additional weight during hunting and climbing. Long fur can become caught in vegetation. This reduces mobility in dense brush. Prey detection may decrease if fur blocks whiskers or vision. These trade-offs explain why long-haired cats are less common in wild populations. Domestic cats face fewer survival pressures. Humans provide food and shelter. The disadvantages of long fur matter less in domestic settings. Laperm cats demonstrate that coat genetics involves multiple genes. Laperms have curly long hair. The curling gene is separate from the length gene. Both genes must be present for the characteristic Laperm coat. This shows that coat phenotype results from interactions between multiple genetic loci. Historical records document the development of long-haired breeds. Persian cats appeared in European records in the 1600s. Early descriptions note their unusual fur. Breeders in Victorian England formalized Persian breeding programs. They selected for increasingly long fur. Show standards developed that favored extreme coat length. Modern Persians have fur 2 to 3 times longer than their ancestors from 200 years ago. This represents rapid change driven entirely by artificial selection. Level 5. Show cats hair follicle cycling operates in 3 phases. Anagen is the growth phase. Cells in the follicle bulb divide rapidly. The hair shaft extends. In long-haired cats, anagen lasts significantly longer than in short-haired cats. Catagen is the transition phase. Cell division stops. The follicle begins to shrink. Telogen is the resting phase. The hair remains in the follicle but does not grow. Eventually the hair sheds and a new anagen phase begins. The FGF5 protein normally signals the end of anagen. When this protein functions correctly, anagen lasts approximately 30 to 45 days in cats. The mutated FGF5 gene produces non-functional or reduced function protein. The growth phase continues for 90 to 120 days or longer. This extended anagen produces hairs 2 to 3 times longer than normal. Keratin is the structural protein of hair. 2 types exist in cat fur. Alpha keratin forms the hair cortex. This provides mechanical strength. The amino acid composition of keratin affects coat quality. Cysteine residues form disulfide bonds between keratin molecules. More disulfide bonds create stronger, coarser hair. Fewer bonds result in softer hair. Selkirk Rex cats produce keratin with altered bonding patterns. This creates their characteristic curly texture combined with long length. Thyroid hormones regulate coat quality. Hypothyroidism causes poor coat condition. The fur becomes dry and brittle. It mats easily. Hair growth slows. Coat quality serves as a clinical indicator of thyroid function. Ragdoll breeding lines show variation in coat quality. Lines with optimal thyroid function produce silky, mat-resistant coats. Lines with subclinical thyroid issues produce coats that mat more readily. Photoperiod influences seasonal coat changes. The duration of daylight exposure affects the cat's endocrine system. Melatonin secretion changes with day length. This triggers hormonal cascades. These hormones signal follicles to enter anagen or telogen. Indoor cats with constant artificial lighting show reduced seasonal variation. Their coats remain relatively consistent year-round. Outdoor cats show pronounced seasonal changes. Cymric cats kept outdoors in northern latitudes develop exceptionally thick winter coats. Breed standards specify coat characteristics with precision. The Persian standard requires coat length of 3 to 5 inches. The texture must be fine and silky. The coat must stand away from the body. Judges evaluate these characteristics at cat shows. Chantilly-Tiffany breed standards specify semi-long coats with silky texture. The undercoat should be minimal. These precise standards guide breeding decisions. Nutritional requirements for coat maintenance include specific amino acids and fatty acids. Cats require dietary taurine. Taurine deficiency causes poor coat quality. Omega-3 and omega-6 fatty acids maintain skin health. These fatty acids incorporate into cell membranes. They reduce inflammation. They support sebum production. Sebum oils the hair shaft. This prevents dryness and brittleness. Highland fold cats with adequate omega-3 intake show superior coat sheen and texture. Protein requirements increase in long-haired cats. Hair is 95% protein. Continuous hair growth demands constant protein availability. Cats with insufficient dietary protein may show reduced coat quality. Growth phase duration may shorten. This partially counteracts the genetic predisposition for long hair. Optimal nutrition maintains the full genetic potential for coat length and quality. Level 6. Foundation breeding cats. Comparative genomics reveals that FGF5 mutations occur independently across felid species. The domestic cat FGF5 mutations differ from those found in some wild felids. Pallas's cat possesses exceptionally long, dense fur. Genetic analysis shows this species has a different mutation in a gene that interacts with FGF5. This represents convergent evolution. Multiple genetic paths produce similar phenotypes. The specific mutations vary, but the outcome remains consistent. Additional loci beyond FGF5 contribute to coat phenotype. Genome-wide association studies identify quantitative trait, loci on chromosomes B1, D1, and E2. These loci affect undercoat density independent of hair length. Karelian bobtail cats show variation in these loci across populations. Island populations demonstrate higher undercoat density than mainland populations. This correlates with harsher winter conditions on the islands. Phenotypic plasticity allows individual cats to modify coat characteristics within genetic constraints. Cats exposed to consistent cold temperatures develop denser coats even within a single season. This occurs through modifications in follicle activity that do not change the underlying DNA sequence. Gene expression changes in response to environmental signals. Cats move from warm to cold climates, show measurable increases in undercoat density within 8 to 12 weeks. Domestication syndrome describes a suite of characteristics that appear during animal domestication. These include changes in coat color, coat texture, ear shape, and tail carriage. The selection for tameness inadvertently affects other traits. Neural crest cells influence both behavior and physical characteristics during development. Selecting for behavioral traits alters neural crest cell migration patterns. This impacts various physical features, including hair follicle development. Domestic longhair populations show greater variation in coat characteristics than wild felid species. This increased variation reflects relaxed selection pressure and domestication syndrome effects. Population genetics analysis reveals the geographic distribution of long-haired trait frequencies. The trait appears at low frequency in random bred populations across most regions. Frequencies range from 3 to 7% in North America and Europe. Higher frequencies appear in cold climate regions. Rural Norwegian cat populations show frequencies of 12 to 15%. Russian random bred populations show similar elevations. These patterns suggest natural selection maintains the trait at higher frequencies in cold environments despite the absence of deliberate breeding programs. Wild felid species show instructive patterns. Snow leopards possess long, dense fur for high-altitude survival. Genetic analysis identifies different mutations from domestic cats. The genes involved in growth regulation differ between species. However, the regulatory pathways overlap significantly. Asian semi-longhair breeds derive from regional populations where wild and domestic cats interbred historically. Genetic markers indicate limited introgression from wild species. This may have introduced alternative longhair alleles into domestic populations. Level 7. Research population cats. Epigenetic regulation of hair follicle gene expression involves multiple mechanisms. DNA methylation at CPG islands in the FGF5 promoter region affects transcription rates. Histone modifications alter chromatin accessibility. Specific histone acetylation patterns correlate with active follicle states. MicroRNAs regulate post-transcriptional gene expression. MIR31 and MIR214 target components of the WNT signaling pathway. This pathway is critical for follicle cycling. Disruptions in these epigenetic mechanisms produce coat abnormalities independent of DNA sequence variation. Environmental factors influence epigenetic markers. Cold exposure alters methylation patterns in genes regulating metabolism and insulation. These changes occur within weeks. Some modifications persist after environmental conditions normalize. This provides a molecular mechanism for phenotypic plasticity. Research on experimental breeding lines demonstrates that maternal environment affects offspring coat characteristics. Dams exposed to cold during pregnancy produce offspring with denser coats. This effect appears even when offspring are raised in warm conditions. The mechanism involves transgenerational epigenetic inheritance. Specific methylation marks transfer from mother to offspring. Developmental timing mechanisms control when and where specific hair types grow. The primary hair coat develops during fetal life. Secondary hair follicles develop in waves during late gestation and early postnatal life. The ratio of primary to secondary follicles determines coat density. Long-haired cats show altered developmental timing. Secondary follicle formation extends over a longer period. More follicles develop per unit area. This creates the dense undercoat characteristic of fluffy breeds. Computational modeling of fur insulation efficiency uses finite element analysis. Models incorporate measured parameters: hair density per square centimeter, hair diameter, hair length, and air permeability. Simulations calculate heat transfer coefficients. Results show that optimal insulation occurs with specific combinations. Very long hair provides diminishing returns above certain lengths. The hair begins to collapse under its own weight. This reduces the insulating air layer. Manole cats possess near optimal hair length for their body size and climate. Their hair length of 4 to 5 centimeters maximizes insulation efficiency. Current research frontiers include hair follicle stem cell biology. The follicle contains multiple stem cell populations. Bulge stem cells maintain the follicle. Dermal papilla cells signal to bulge stem cells. These signals initiate new hair growth cycles. Understanding these signals could enable manipulation of coat characteristics. Research groups investigate whether activating specific signaling pathways extends anagen phase further. This could produce even longer hair without genetic modification. Unresolved questions persist regarding the full genetic architecture of coat variation. Genome-wide association studies identify significant loci but explain only 60 to 70% of phenotypic variation. The missing heritability may reside in rare variants not captured by standard genotyping. Structural variants such as copy number variations may contribute. Epistatic interactions between genes likely play roles. Identifying these factors requires whole genome sequencing of large populations with detailed phenotype measurements. Cross-species comparative studies examine coat characteristics across all felid species. Snow leopards and mantle cats represent independent evolution of long fur. Comparing genetic differences between these species and short-haired felids identifies convergent genetic changes. Such changes likely represent essential pathways for long hair development. Studies also compared domestic cats to ancestral wild populations. Genetic analysis of ancient DNA from archaeological cat remains shows when long hair mutations first appeared in domestic populations. We started with a simple question: what makes a cat fluffy? We looked at this question 7 different ways. We saw pet cats with soft, thick fur. We learned about common fluffy breeds from around the world. We studied regional cats that adapted to cold climates. We examined purebred cats with specific coat standards. We explored championship cats bred for perfect coats. We analyzed wild and rare cats with unique fur patterns. We finished with laboratory cats that help scientists understand genetics. Each level showed us something new. The fluffy coat is not just one thing. It comes from genes, environment, breeding, and evolution. Scientists continue to study cat fur. New discoveries happen every year. The next time you see a fluffy cat, you will understand more. You will see the undercoat and guard hairs. You will think about the genes that made that fur grow. You will recognize the adaptations for cold weather. You will appreciate the complexity hidden in something that looks simple. Fluffy cats carry millions of years of evolution in their coats. That is what makes them remarkable.